Hello, all the KVPY aspirants. I welcome you to the second video lecture of biology for KVPY. In this video lecture, we are going to discuss the part 2 of 2019 KVPY paper, in particular stream SA. So SA, the paper which is meant for class 11 students. We are going to discuss the part 2 biology of this paper in this video lecture. In the previous video lecture, we have already covered the part 1 of this particular paper. Okay. So without wasting a lot of time, let's dive in. And the very first question of part 2 is question number 76. Okay. So before starting, let me add that this particular section is comprised of only 5 questions. So part 2 has lesser number of questions. However, each question is of 2 marks. Okay, so pay attention closely. If you see here, every question is of 2 marks. So part 2 is the questions of part 2 of all the subjects are more difficult problems. They require more thinking and hence they carry more marks. Okay, great. So let's start. If we see here, the question number 76 says that a cell weighing 1 mg. Okay, let me zoom it for you. Okay, so I hope it is readable to every one of you, even for the students who are reading it or watching this video on the mobile phones or devices having relatively smaller display. Okay, so that should be readable to everyone. Well, so this question says that a cell weighing 1 mg grows to double its initial mass before dividing into 2 daughter cells of equal mass, right? Correct. So far the statement 1 is very simple. It says that there is one cell which is of mass 1 mg, correct? So you have some information here. It is saying that one cell is equals to 1 mg, and when it is fully grown and about to divide, its mass increases to 2 mg, and then it again divides into 2 daughter cells, each having 1 mg. So relatively or ultimately what you find is there are cells of each having 1 mg mass. Great. Let me tell you, it's quite an exaggeration. The individual cells are not of 1 mg mass. Okay, it is an exaggerated number, but it is given, it is data, so we are taking it. Okay, great. What is the next statement saying, next line saying? Assuming no death. Okay, so we are not going to assume any death. At the end of 100 divisions, what will be the ratio of the mass of the entire population of the cells to that of mass of the earth, right? So see, the question is asking for a ratio, right? Ratio of mass of these bacteria, right? Mass of these bacteria upon mass of the earth. Great. ठीक है. So if you look at यहाँ पे cells दिया हुआ है, actually they mean to say bacterial cells, that's why I have written bacteria. You can write cells as well. So the question is asking for the ratio of the mass of these cells upon mass of the earth, right? Now in this question there are some facts given, right? And this fact is they have given the mass of the earth. Great. And the mass of the earth they have given to be 10 to power 24 kg. Okay. Also they have given an approximation that 2 to power 10 is equal to 1000. Okay. In reality, let me tell you, in reality 2 to power 10 is equal to 1024. Okay, but this question is asking us to use 2 to power 10 value as 1000. So again, a rounding off. we will use this for the calculation in this particular problem. Okay, now so if we have to take this ratio, if we have to calculate this ratio, what we are going to do, we need mass of this bacterial cell, right? And we already know from this question that one cell is equals to 1 mg, correct? So the total number or the mass of all of such cells will be number of such cells. Okay, I am taking it to be n here. Okay, sorry, not n, let us take it to be x into multiplied by mass of each cell, correct? That will give you the mass of all of such cells upon mass of earth, that is 10 to power 24 kg, right? I hope there should be no problem in this. Now let us see. So number of such cells, I have taken x into mass of each cell. I know it is given 1 mg upon mass of earth has been given 10 to power 24 kg, right? Since the upper unit is in mg, we are going to convert kg into mg. So if you convert kg into mg, you have to multiply it by a factor of 10 to power 6, right? Because 1000 milligram is 1 gram, and then 1000 such grams will form kilograms. So 10 to power 3 into 10 to power 3, that is 10 to power 6, correct? So this much milligram. Now we can take the ratio because both the numerator and denominator are in the same unit, correct? So mg will be cancelled, we will see later. So in order to take this ratio, all we need is the value of x, correct? So in this question, you actually have to calculate the number of such cells. Okay, now if you look at the question once again, they're saying at the end of 100 divisions, right? At the end of 100 divisions. Okay, so they have given this data to calculate the number of such cells. Okay, well, they have also said that they have started with one cell, right? Now they have started with, initially there was one cell, and they are asking that after 100 divisions, what will be the value of x? That is number of cells, assuming that there is no death. Well, this is the real problem here. You have to calculate the number of cells. Now, we know that the growth equation for such cell is 2 to power n. They show exponential growth, right? Why it comes so? See, one cell will divide to form 2 cells, then these 2 cells will divide to form 4 cells. See, 1 will divide into 2 cells, then both these cells will divide 4 cells, then they will divide to form 8 cells, correct? Can you see? So they are following, so 2 to power 0, 2 to power 1, because there is one division. This is first and this is second division, 2 to power 2. This is third division, 2 to power 3. See, 8, 4, 2, 1. So this whole division, the rate of growth of the cells follows the rule of 2 to power n. That is exponential growth. Okay, so they show exponential growth. They reproduce by 2 to power n rule. The number of such cells or such unicellular organisms divide or they grow by a factor of 2 to power n, where n is equal to number of divisions. Okay, so if you know the number of divisions, you can easily calculate the number of cells. Well, here we know that we started with a single cell, so 2 to power n, n is what, 100 divisions they have given. So if we calculate 2 to power 100 value, this will be the value of x. I hope there is no doubt in this. Great, let's move ahead. What we can write? 2 to power 10. 2 power 10 will be 2 to power 100, right? Or we can write it as 2 to power 10 is given 1000 to power 10, right? Great. Now, what you can write here is 1000 can be written as 10 to power 3, okay? To power 10, that is 10 to power 3 into 10, that is 10 to power 30, right? So the value of x is equal to 10 to power 30, or we can write it 1 into 10 to power 30 cells. Okay, so what we have got is after 100 divisions, one single cell will end up forming 10 to power 30 cells. Okay, so after 100 divisions, from one cell there will be 10 to power 30 cells formed, right? Great. So we have calculated the number of cells, right? So if we have number of cells, we also have the mass of such cells. So mass of all such cells will be equal to 1 into 10 to power 30 into 1 mg. So obviously it is equal to 1 into 10 to power 30 mg. Now we have mass of all such cells and we have mass of earth that was given. We can have the ratio. Great. So ratio would be 10 to power 30 mg, that is mass of all such cells, upon 10 to power 30. Remember 10 to power 24 kg was the given mass of the earth. If we convert it into mg, it will be 10 to power 30 mg. Cancelled. Obviously the ratio is 1. So what we have calculated, the ratio is 1. So the answer would be option C. Great. Now, what is the motive behind such a question? The motive is, such question is trying to teach you that the bacteria or unicellular organisms grow by an enormous speed. Okay, and if they are allowed to grow at their full potential and there is no mortality, then in no time they can supersede even the mass of the earth. Okay, great. Now let me also tell you that the rate at which they divide, that is the doubling time of bacteria, could be in minutes. Say for example, for E. coli it is just 20 minutes. Okay, so these unicellular organisms or bacteria divide at an incredible speed. Well, so this was the first question. Now let's move on to the next question, that is question number 77. Well, the question number 77, right? So let us read this question first. This question says, papaya is a dioecious species. Okay, it is a dioecious species with XY sexual genotype for male and XX for female, right? So this type of sexual phenomena we see in humans as well, right? Where if the organism has XX chromosome, then it is female, and if it has XY, then it is male, right? I hope you all know this. So similarly, they are saying that papaya, in case of papaya plant, which is a dioecious plant, that is, in papaya, one plant will be male plant, the other will be female plant, right? It is dioecious, that is, both the sexes are present on two different bodies. Dioecious, right? See, this word di means two and cs means body. Okay, so whenever there is dioecious, it means that the organism have male and female sexes on two different bodies, right? Male and female sexes are not present on the same body. They are present on two different body. Okay, that is, you will find male and female, different individuals, as we see in case of humans or other animals, right? Dioecious. or dioecious is also, you can consider it as unisexual, right? So they are saying that papaya is a unisexual plant, which is true. Okay, this happens in case of papaya plant. One will be male plant, the other will be female plant, and male plant will only bear male flowers, female plant will only bear female flowers. Okay, so if you talk about the flowers of papaya, they will be incomplete flowers, right? Great. And you find that the male and female distinction is at the genetic level, and the female have XX chromosome, the male have XY chromosome. It is similar to humans. Great. Now the question says, what will be the genotype of embryos and endosperm nuclei after double fertilization? Okay, so the question is asking about the genetic makeup or genotype of the embryo and the endosperm. Okay, endosperm or endosperm nuclei, you can consider them the same thing. Okay, great. So endosperm nuclei after double fertilization, you know that in plants double fertilization occurs. Not all the plants, only in case of angiospermic plants. Okay, now what is this double fertilization? We are going to see here in brief, but if you want a detailed information on double fertilization, I would recommend you to go and see the video on double fertilization, which is there on our channel in class 12. Okay, so when you go on our channel in class 12 biology lectures, you will find a particular lecture on double fertilization. It is a topic of chapter number 2 of class 12, that is reproduction in flowering plants. Okay, so I would recommend all of you to go and see that video for detailed information on double fertilization. Okay, great. However, in this class, let me show you a brief of double fertilization, right? So what is double fertilization? Let me check. So let me show you, not check. Double fertilization. Okay, now can you see this word itself is saying that there will be 2 fertilization events, correct? So in case of angiospermic plants, when they undergo the process of fertilization, there are 2 simultaneous fertilization events. Okay, usually in most of the animals or other organisms except angiospermic plants, there is a single fertilization event. Okay, that is the 2 gametes will fuse. Okay, so the fusion will be a single event, so single fertilization event occurs. But in case of angiospermic plants, simultaneously 2 fertilization events occur. What are these? See, first fertilization event is called syngamy. Okay, and in this the male gamete fuses, okay, with the female gamete. Okay, with the female gamete to form zygote, and this zygote later on will form the embryo. Okay, then what is the second fertilization event? The second fertilization event is called triple fusion. Okay, why we call it triple fusion? Because in this fertilization event, actually there are 3 nuclei which fuses together. Okay, so there are 3 nuclei that fuses together. What are these 3? 2 nuclei n plus n of the central cell. Okay, of the central cell, and 1 nuclei from the male gamete. Okay, so in case of triple fusion, the 2 nuclei which are present in the central cell, okay, these 2 nuclei fuses with the 3rd nuclei of the male gamete. Okay, so 3 nuclei are fusing together. That's why this event is called triple fusion. As a result, PEN is formed. Okay, what is PEN? PEN is called primary endosperm nucleus. Okay, so it is primary endosperm nucleus. Now, if three haploid nuclei are fusing together, then what will be the ploidy level of PEN? Obviously 3N. Okay, therefore the endosperm formed in angiospermic plant as a result of triple fusion, or you can say the double fertilization, is triploid. Okay, so triploid is formed, and later this PEN develops into endosperm. Okay, therefore what will happen? The nucleus of each endospermic cell will be triploid, 3N. Is it okay? Now why it happens so? Let's see. Because if you see the ovule of angiospermic plant, what you find here is egg cell, and there are two synergids. Okay, this forms the egg apparatus, and on the chaliceal end there are three antipodal cells. Okay, and this part, this central part, is a single cell that is called central cell, and this has two haploid nuclei. Okay, now when fertilization happens, okay, when fertilization happens, that is when the pollen tube comes inside. Okay, at the time of the entry, okay, what happens when it enters? It enters through one of the synergids. So these two, which forms the egg apparatus, are called synergids. The two on the either side of the egg cell. This is egg cell. Okay, when it enters, by that time this synergid and this synergid have degenerated. At the same time, these antipodal cells have also degenerated. Now this pollen tube brings in two male nuclei with it. Okay, these are two male gametes. Okay, so this is one and this is second. Now this one male gamete fuses with the egg cell to form embryo, while the other go and fuses with these two nuclei of the central cell to form PEN, primary endosperm cell, which later forms the endospermic nuclei or the endosperm. Okay, so let me show you here what will happen. These two red nuclei and the second male gamete, these all will fuse to form PEN, which will be triploid, and these two will fuse to form these two will now fuse to become a single nucleus. This will be zygote, which will be diploid. Okay, so this is in brief about the double fertilization. For more details, you should go and watch that video. There we have spoken in great detail about how this process happens, what is the overall structure of the embryo sac and the ovule, how this pollen tube enters into the ovule, every detail of it. Okay, great. So I strongly recommend to go and watch that video. Now what happens here is, this is forming the endosperm and this is forming the embryo, right? And this is what is being asked in this question, that what will be the genotype of the embryo and the endosperm. Great, let us see. When we are talking of embryo, it is formed because of the fusion of the male gamete and the egg cell, that is female gamete. Okay, female gamete, which is egg cell. Now you know that the female will have X chromosomes. So when this will undergo meiosis, this cell is diploid, right? When this will undergo meiosis to form the egg cell, the egg cell will have X. Okay, the egg cell will always have one X chromosome, correct? So this will have only X. The male gamete will be formed from the male, and the male has X and Y both chromosomes. So one male gamete may have X chromosome, the other may have Y. Correct? So the pollen grains produced by a plant may have X chromosome or may have Y chromosome. So the male gametes formed here may have X or Y. Now, if a zygote is formed because of the fusion of X type of male gamete, then the embryo formed will be having XY chromosome. If not, if it fuses with the X type of male gamete, then it will be having XX chromosome. Correct? Right? Now, let's see. If we are talking of male gamete, there is 50% chance that a pollen grain will be having X chromosome and another 50% chance of having Y chromosome. And therefore, the zygote formed having XX chromosome will have 50% chance, right? Similarly, zygote having XY will have 50% chance. Is it clear? Therefore, the embryo resulting from such zygote, this is zygote, okay? This embryo, 50% embryo may have XX, and this 50% embryo may have XY. Okay, so in case of double fertilization, 50% of embryos may have XX chromosome and another 50% have XY. What we are trying to say here is there is 50% chance that an embryo formed will have XX chromosome. Okay, similarly, it have another 50% chance that it may be XY. Correct? Good. What about the endosperm? If we are talking about endosperm, we have just seen that endosperm develops because of fusion of N plus N, two nuclei present in the central cell, and this central cell is part of female gametophyte, right? So they will, both of these N will have X chromosome only, and then it has to fuse with the second male gamete, and this male gamete has 50% chance of having X and another 50% of having Y. Correct? Therefore, if the fusion is happening, suppose this has fused, this fusion is occurring between this central cell and the second male gamete, there is 50% chance of forming XXY and another 50% chance of forming XXX. Isn't it? So there is 50% chance of endosperm nucleus having XXY and another 50% chance of having XXX. Great. What we have seen so far, that if we are talking of embryo, 50% chance is that the embryo will have XX and another 50% of having XY. In terms of endosperm, 50% chances of having XXX and another 50% of having XXY. समझ में आ रहा? Now let's go back to the question. So when we look at the question, the options are 50% ovules will have XXX endosperm and XY embryo. Okay, while the other 50% would have XXY endosperm and XX embryo. Okay, ठीक है, चलिए next option देखते हैं. You all should always read all the 4 options before choosing your answer. Okay, this you have to do. If you do not, you are bound to make mistakes. Okay, so for all the aspirants who are aspiring to crack any kind of competitive examination which includes MCQs, I really recommend to read all the four answers carefully, sorry, all the four options carefully before choosing one. Okay, do not make blind mistakes. You read the first option and you say that this is the correct one. Don't do that. Don't rush for it. Okay, if you are rushing for it, you are bound to make mistakes. Great. So we are not going to make that mistake. We are going to read all the four options carefully. Okay, especially part 2, which is trickier, because and hence they carry 2 marks, so we can't lose that. Let's see. The B option says 100% ovules would have XXX endosperm and XY embryo. Now we straightforward know that this is a wrong option, right? Great. C, 100% ovules would have XXY endosperm and XX embryo. No, this is also wrong. You know that, correct? There's 50% chances of either of the genotypes that we have seen right now. D option says 50% ovule would have XXX endosperm and XX embryo, while other 50% would have XXY endosperm and XY embryo. Well, now here is the confusion coming up, right? So if you closely see the option A and option D, both are saying of 50% chances. Then where is the difference? Now look carefully. Option A says that the 50% ovules would have XXX endosperm and XY embryo, right? And other 50% would have XXY endosperm and XX embryo. D says that 50% ovule would have XXX endosperm and XY embryo, while other 50% would have XXY endosperm and XY embryo. Can you see, mark the difference? Difference is in the combination. Can you see? So the D option is saying that when the endosperm having XXX genotype will be formed, then the corresponding embryo of that particular double fertilization will have XX genotype. Okay, and if the endosperm has XXY, then the embryo will be XY. Can you see? Now, in order to answer such a tricky question, you need to know the process of formation of these male gametes in good detail. Okay, let us see. So, see what happens. Be careful here. You know that in case of papaya male plant, it has XY chromosome, right? So the pollen mother cell will have XY and it is diploid. When this will undergo the process of meiosis, it will form 4 cells, right? 4 cells, and these 4 cells, 2 will have XX, the other 2 will have only YY, correct? Now, each one of these microspores, these are microspores, will develop into pollen grain. So this will become one pollen, this will become another pollen, this will become another pollen, this will become another pollen, right? So this pollen will always have X, X, Y, Y, correct? So this is pollen grain. Now once this pollen grain is transferred to stigma, that is pollination will occur. Now on stigma, it will further undergo the development, right? And this pollen grain will give rise to 2 male gametes through mitosis. Okay, so this is going to form the 2 male gametes. If this male gamete is formed from this pollen grain, both the male gametes will have X and X. So first and this is second male gamete. Okay, if this will form again XX, if this will form, both the male gametes will have YY. Okay, and if this form again YY. What you find here is both the male gametes will have the same genetic makeup. They can either have X chromosome or both will have Y chromosome. Getting it? Yes. So now see here. If in a double fertilization case, here is the ovule and here you have the egg cell. Egg cell have X, right? So egg cell I'm putting out here, it has already X, one X, and now it is fertilized with the first male gamete. Okay, and here is a central cell having two nuclei. Okay, this will be having X plus X, correct? Now this will be fertilized with second male gamete. Now if the male gamete, this is a male gamete having Y chromosome, then both of them will have Y chromosome. Okay, so this will fertilize with Y chromosome, leading to formation of a zygote having XY, and this will also then fertilize with a male gamete having only Y chromosome, so this will result in XXY, correct? So 50% chance is that the endosperm will be XXY and XY embryo, right? If this male gamete has X chromosome, then what will happen? Then there will be another 50% chance, that is X plus X, so XX, and here will be, can you see? What I'm trying to say here is, if the pollen grain having Y chromosome will come and fertilize, the chances of which is 50%, in that case XY endosperm and XXY endosperm and XY embryo will be formed. Okay, if in another 50% chance the male gamete having X chromosome comes and fertilize this ovule, then in that case the another 50% will be triple X endosperm and XX embryo. Okay, getting it? So if in a seed there is an embryo having XX genotype, then the endosperm will always have XXX genotype. Okay, if in a seed or in an ovule the zygote is XY, then the endosperm will be XXY. Okay, now having this information, what we find, the correct answer would be D. The correct answer would be D. Okay, so if you don't know how the male gametes are developed, then you are unable to answer such questions. Therefore, I always recommend that go and read the biology in good detail. Okay, you can't just read it superficially. If you do so, then such questions will trick you. Okay, great. Fine. So let's move on to the next question. Well, so moving on to question number 78. Okay, this question says that solid and dotted lines represent the activity of pepsin and salivary amylase enzymes of the digestive tract respectively, right? So the solid line here represents the pepsin, okay, and the dashed line represent the salivary amylase. Okay, great. Next line says, which one of the following graph best represents their activity versus pH? Okay, so what this question is asking you to pick the correct graph for the activity of these two enzymes versus their pH. Okay, now before attempting this question, let me give you some required information. Now, what is this information required? You know that pepsin, okay, pepsin is a proteolytic enzyme. So it works on the proteins and partially digest them into proteoses and peptones. Okay, this you must have studied in your standard 10th first chapter life processes, where you study about nutrition. Okay, so in standard 10 you have already read it. You have studied about pepsin, and you know that it is secreted in the gastric juice. So it is secreted by the gastric glands, that is the glands present in your stomach, okay, and they form the gastric juice. And the pepsin, being part of the gastric juice, it is present in an acidic environment, right? Remember, the pH of gastric juice was around 1.8 or 1.8 to 2.5. Remember? Great. Now the pepsin is such a proteolytic enzyme that acts only in acidic condition. Okay, the optimum pH, okay, the pH at which the pepsin functions with its optimal capacity or maximum capacity is around 2. Okay, so near to 2.0 pH, pepsin works efficiently. Right? Similarly, let me tell you that salivary amylase is a type of amylase, okay, and all amylases are carbohydrate digesting enzymes. So that is, they act on carbohydrate and degrade them either partially or completely into simple sugars. Okay, great. If they are degrading the carbohydrate partially, then they will degrade it into oligosaccharides, okay, or maybe disaccharides. But if they are doing complete digestion, then they will digest the carbohydrate into simple sugar like glucose, fructose, galactose, and so on. Okay, see, now salivary amylase is part of saliva, right? It is secreted by the salivary glands and it forms the saliva, and the pH for saliva is 6.8. Is 6.8. It is slightly acidic, just 0.2, okay, units lower than the neutral pH of 7.0. Correct? So it is slightly acidic. Now salivary amylase is or works best in slightly acidic condition, that is about 6.8 pH. Okay, so what we can say is the optimum pH for salivary amylase is 6.8. Now these two pieces of information you have, right? Apart from that, let me tell you one more thing. If we are talking of enzymes, okay, if we are talking of enzymes, we find that every enzyme works best in a certain condition. Okay, if the conditions are not proper for that enzyme, then it will not work at maximum capacity. Okay, now if we are talking in terms of pH, so on the y-axis we are having the enzyme activity here. Okay, so the pH at which the enzyme has maximum activity is called its optimum pH, right? What about the other pH values? As you go away on either side of this optimum pH, slowly the enzyme activity decreases. Okay, and after a particular pH values, the enzyme activity becomes 0. Okay, so whenever you draw an enzyme activity graph, what you find, it forms a bell-shaped curve, something like this. Okay, so this is the pH at which this enzyme is having maximum activity. So this particular pH is called optimum pH for that enzyme, and as you are going on the either side of this optimum pH, slowly the enzyme activity is decreasing, right? Are you getting it? So for any enzyme activity curve, it forms a bell-shaped curve, right? Great. Now you have the necessary information to solve this question. Let us see now. Okay, I'm deleting it so that we can have space. Great. So see, graph A, both the curves are kind of enzyme curves for pH. Great. If you look at B option, it is actually inverted bell. So obviously this is incorrect. This will not happen. Enzyme kinetics do not form such graph. If you see C and D, well, C and D are also not the typical enzyme curves. So this is also wrong. If you see the A option, see pepsin has a optimum pH of around 2. See, this was 5. So this would be 4, 3, 2. Can you see? So around 2 point something is the optimum pH here shown for pepsin. As you deviate on the either side, the activity is decreasing. Similarly for salivary amylase, it is around 6.8 or close to 7. Okay, slightly lesser than 7. So if you are deviating again, the activity is decreasing. So the graph that best depicts the enzyme kinetics or enzyme activity curve for pepsin and salivary amylase would be A option A. Great. Let's move on to the next question. If you want to know more about this enzyme curves, we study in class 11 when we talk about the chapter biomolecules. So there is a section of enzymes where we study about the enzyme kinetics. Okay, and there we study how the enzyme functions, what are the conditions required, role of inhibitors. We derive the Michaelis-Menten equation which governs the enzyme kinetics. Okay, so this all we will study in 11th under the heading biomolecules. Okay, great. Well, moving on to the next question. Let's look at the question number 79. Okay, question number 79. This question says that if the gene pool of the locus X in the human genome is 4, then what would be the highest possible number of genotypes in a large population? Okay, now this is the question of genetics, and there will be more of maths involved in it. Okay, let us see. Let us first try to understand this question. So you know that most of the organisms are diploid. They are diploid. Great. Diploid means inside their nucleus every chromosome is present in a pair. Okay, that we call homologous pair. So suppose this is chromosome number 1, so this will be 1 prime, right? This is the homologous pair. These are 2 chromosomes which are similar, not identical, they are similar. Okay, they have the same type of genes, not the same alleles, but the same genes at each locus, same number of genes, same type of genes, same arrangement of genes, and therefore the size, shape, function, everything of these 2 chromosomes will be almost same. I'm not using the word identical, they will be same. Okay, and that's why we call it one homologous pair. Okay, let me tell you that homo means same and logos is for locus. Locus means location. Since these 2 chromosomes have the same locus, that is at their locus same type of genes are present, same genes are present, that's why we call such chromosomes homologous pair or homologous chromosomes. Okay, one of this comes from father, the other comes from your mother. Okay, great. Now the question says that suppose at one locus, so I am taking here this as one locus. Okay, so this as one locus, and this locus in this question has been named as X. Okay, so this is, let me zoom it. So this locus has been named as X, and they are saying at this locus there are total of 4 genes. Correct? See, the genome is 4 genes, right? Then what would be the number of possible genotypes in a large population, right? Now, let us see. Suppose there are 4 alleles, right? I'm saying A, B, C, and D. These are 4 alleles or type of genes present at this locus X, right? Now, we know, or considering that the organism we are talking here is a diploid organism, any organism will have 2 of these genes, right? So that could be either AA or AB or AC or AD, or it could be BB, BC, BD, or it could be CC, CD, or it could be DD. Are you getting it? Other than this, there is no other combination possible. Okay, so the total number of genotypes or type of genotypes that you can find is, see here, 4 plus 3 plus 2 plus 1. Getting it? Now, for your better understanding, this will be equal to how many? 7, 9 and 1, 10. Are you getting it? So for this question, the answer would be 10. Okay, but that is simply the answer. Let us talk something more about this question or such question. Suppose there would be 5 such genes, then what would have happened? A, B, C, D and E. In that case, again, the possible combination here would be AA, AB, AC, AD and AE. Total how many? 1, 2, 3, 4 and 5. BB, BC, BD and BE, 4. Right? Similarly here, CC, CD and CE, here that will be 3. DD and DE, 2, and here only EE plus 1. That would make it 15. Right? Now, so can you see that this whole equation, if you see here mathematically, it is a arithmetic progression, AP. It is following arithmetic progression. Can you see here? 1 plus 2 plus 3 plus 4 plus 5. Okay, it is increasing by a factor of 1, so common difference is 1, right? So it is forming a AP series. Now, if you know little bit of mathematics, you know that the formula for AP series is n by 2 into 2a plus n minus 1 into d. Correct? So this is a general formula for AP, where n is the nth term, a is the initial term, and d is the common difference. Right? Some of you may be knowing this already, some of may not. If not, nothing to bother. Okay, do not panic here. That why am I teaching mathematics in a biology class? Do not panic, we will simplify it further. Okay, now in case of biological systems, we always start from number 1, and there will be the common difference, that is the value of d would be 1. So considering that in biology the value of a and d is equals to 1 always, this equation reduces to n by 2 multiplied by 2 into 1 is 2 plus n minus 1. I should write like this. Correct? We can further reduce this formula into n by 2 multiplied by n plus 1, right? Minus 1 plus 2. Okay, so n by 2 into n plus 1. So this will be the formula. Okay, so in such a question, if the question says there are 10 genes at that locus. How will you calculate? Put the value of n to be 10. What will be your answer? 10 by 2 plus into 10 plus 1, that is 5 multiplied by 11, that is 55. Okay, now let us take some examples from the biology that we study, okay, that we find in human body or human physiology or human genetics. Let us see. Suppose from this formula, if I say that there are 3 alleles for a gene, okay, so n value will be 3. In that case, the total number of possible genotypes would be, let us calculate, n value is 3, so 3 by 2 multiplied by 3 plus 1. How much? 3 by 2 multiplied by 4. 2, 3 2s are 6. Great. Now you will say, where we see it? We see it in case of blood type inheritance in humans. You know that humans have ABO type blood group. Okay. And there are 3 alleles responsible for it. And these alleles are small I, capital IA, and capital IB. Okay. Now, what is the possible genotypes? The possible genotypes would be IA, capital IA, or it could be small I, capital IB, or could be IA, or IB, or it could be IB. Is there anything else possible? No. Right? So if you see, if a person has both the alleles as II, the blood group would be O. If small I and capital IA, A, B, again A, B, and in this case AB. Getting it? So the total number of possible genotypes, how many? 1, 2, 3, 4, 5, and 6. Can you see? This formula is correct. 6. Now let me give you one more familiar example for you. Suppose there are only 2 alleles for a gene, as you have studied in standard 10th for the pea plant, your favorite genetic experiment, TTE, right? So suppose there are 2 alleles, that is T and small t, capital T and small t for a gene, correct? So there are only 2. In that case, from our formula, if n is equals to 2, the possible number of genotypes would be 2 by 2 multiplied by 2 plus 1. So cancelled, 2 plus 1, 3. Let us see how it happens. If you have 2 alleles, the 3 possible combinations that you already know is capital T, small t, and capital T, small t. So these are the 3 possible genotypes. Are you getting it? Correct? So this is how you calculate such problems. And to calculate such problem, the equation that you need is this one. So total number of possible genotypes would be n by 2 into n plus 1. Please remember this. This is the shortest formula for you. And those of you who are phobic to mathematics, who do not want to study of AP and other mathematical calculation, do not panic. It is very easy. In most of the biological problems, the number of alleles or genes present at a locus will be not more than 6 or 7 in your question at your level. Okay, and you can simply add the numbers like 1, 2, 3, 4, 5, 6, 7 and get your answer. Okay, as we have done earlier, for 5 it is 15, for 4 it is 10, and so on. So if you don't want to remember this equation, it's okay. Just remember you have to add 1, 2, 3, 4 up to the number of alleles or genes that is given at a particular locus, or the number of alleles found in a gene in a population. In either cases, the approach and the method to solve the question would remain same. Okay, so the answer will be 10 here. Great. याद रहेगा? थोड़ा सा मैथमेटिक्स हमने किया यहाँ पे. It is a arithmetic progression series. Great. So now let's move on to the next question. Well, the next question is question number 80, which happens to be the last question of part 2. Okay, and also the whole paper of SA 2019 KVPY. Well, so this will be the last question. Let's see. The question is a match the following. You have to match the two columns, and this question has been asked from the phytohormones. Okay. Well, if you read out the question, what we find, the column A has PQRS and then column 2 has 1 to 5. So column A has only 4 items, whereas column 2 has 5 items, and this is to confuse you. Okay, let's see. Now what they're saying is abscisic acid, ethylene, cytokinin and gibberellin. These all are phytohormones. Apart from these 4, there is a 5th phytohormone called auxine, which is not there. Great. Lesser the number of hormones or the items in the master column, lesser is your problem. Okay, great. Column 2, the first one says promotes disease resistance. Second says maintains seed dormancy. Third says promotes seed germination. Fourth says promotes fruit ripening. And fifth says promotes leaf senescence. Okay, now we have to match them. Well, let us start. Now, whenever you have such match the columns or match the following type of problems, there is a trick, okay, to make the things faster for you. You look at the options first, okay, and see that which of the options has some common, okay, something common. So you can see here that for Q, 4th option is common here, right? The rest, nowhere you find anything common. Can you? No, nothing is common, right? It means that Q is for 4. The Q is going to match with 4. Now, let us go see and match with our theoretical knowledge. Q is saying that ethylene, and it matches with 4. So ethylene is matching for promotes fruit ripening. Now it is true. It is true. The main function of ethylene, which is a gaseous phytohormone, is that it promotes fruit ripening. Okay, so this is correct. Now on this basis, you have already rejected the 2 options, C and D. So the answer could either be A or B. Now if you know the answer of P, then you have the answer, right? P says abscisic acid. Now abscisic acid is saying 3 promotes seed germination. No, it is wrong, right? It is saying maintains seed dormancy. Yes. So abscisic acid maintains seed dormancy. This is one of the function of abscisic acid. Okay, it is a plant growth inhibitor. Abscisic acid is a plant growth inhibitor, and therefore it inhibits the overall growth, and hence it will help in maintaining the seed dormancy. Okay, now what is this? Again, to understand about seed dormancy and things like that, you can go and study some of it from class 12th video lectures that is part of reproduction and flowering plants. Okay, so in class 12 second chapter we have made some videos where we have spoken of seed dormancy and things like that. You can go and refer to them. Okay, as of now these all you may have studied in class 10th as well. Okay, so in class 10th under the chapter control and coordination, you have to study about the role of all the 5 phytohormones. Okay, so either you recall from there or you can study in your class 11 when you study of plant physiology. There also we talk about all this thing. This is a part of your syllabus, class 11 syllabus. So abscisic acid maintains seed dormancy. So obviously the answer would be second. Let us see rest of the matches as well. So R is matching with 5th, that is R cytokinin is matching with 5th. Inhibits leaf senescence. Now cytokinin is a plant growth promoter. It mainly promotes the cell division. It promotes the mitosis in plants, right? And this inhibits the leaf senescence. It inhibits the premature senescence of leaves or flowers and fruits. Okay, so this match is also correct. We know it. Gibberellin. Now, gibberellin is known to promote the seed germination. Okay, in fact, it is used in horticulture and other farming techniques. It is used for inducing the seed germination. Okay, especially in brewing industry, it is used. Great. So the answer is B. Fine. To tackle such question, you need to have some theoretical knowledge of all these hormones. And where you can find them in your NCERT book, class 11 NCERT book. If you open and go in plant physiology section, I think it is unit number 4. Okay, there you will find detailed function of all the 5 phytohormones. I would strongly recommend to study and learn from NCERT book. Okay, most of the questions regarding these phytohormone functions are directly asked from the NCERT, whether it is KVPY or the NEET. Okay, so go and learn them, and such question will take no time. You can do it fastly. Okay, remember the trick for match the following: always find for the common things. Okay, the commonality between the options. Well, so this completes the part 2 of the biology section for SA 2019 KVPY paper. We have already covered part 1. This was part 2. So we have covered the SA 2019 KVPY paper. Now from next video lecture, we will start with maybe 2018 paper. Okay, so stay tuned and keep studying. If you have any doubts, any problems, or anything else apart from these topics that you want to ask, you can directly call me, or you can reach out to our team, or you can leave your problem in the comment section. Okay, so keep geared up. Okay, no competition is hard. Actually, it's your studies, your preparation that makes it hard or easier. Okay, so prepare yourself and you can really do it. Great. So with that, we will meet in the next lecture of this series. Till then, please remain safe, sanitize yourself, and keep studying. Thank you.